name is Marcus Kelly. I'm a postdoc in Trey Ideker's lab at UCSD. One of the big problems with cancer genomics is that every tumor is unique. But it's also true that we all recognize all of these genetically different diseases as cancer. These different mutations are affecting some pathways in more or less the same way, even though they happen in genes that are on totally different chromosomes. And lots of people use this kind of approach to help us find new cancer genes or new ways of developing therapies and things like that. But the question of what are all of the different pathways and complexes that are consistently perturbed by cancer mutations is not really a question we've thought about in those terms. Like, we think about it on small scales for somebody's project and not the whole cell. But with the CCMI starting to put together the data to actually make these whole cell maps, that puts this very broad question within reach. And so part of my projects are working on modeling the cancer genomes just in terms of the complexes and pathways that help us best understand the cancer genome, with the goal of making cancer genomic analysis simpler, faster, and more informative. The project exists in the form that it does because there needs to be a stepping stone between the interaction mapping experiments that, for example, the Krogan lab does, and the more clinic adjacent work that other members of the Ideker lab do. When you are making an analysis about a particular patient or a bunch of patients, you need that to be in terms that you can understand. Right, and there are other people in the Ideker lab who are working on visible machine learning algorithms that that basically can justify their decisions in terms of cell biology. But in order to do that, they need to know what aspects of cell biology they ought to be caring about in the first place, and that's actually a small subset, of the cell as a whole. And so, my project is positioned as this intermediate between these cell mapping efforts and these patient-facing ones. And so, it's collaborating on both ends in that way. Honestly, one of the big ones stems from, me not collaborating and talking to other people when I should have. And I had made, frankly, a lot of progress doing what turns out to have been faulty statistical methodology. But having caught that, that actually pointed me onto a new and actually totally different path I hadn't taken before, that is the one that we're going to be running with. I guess in some sense that's kind of a negative lesson. I should have been talking to more people earlier. But that said, once I had tuned into the fact that this error was happening, these other people helped set me on the right path. Yeah. This bioinformatics project that I've been doing, is right now it's in a stage where what I'm going to be publishing is the methods, justifying how we got there. But it is a really exciting tool, and even just this weekend, as I've been presenting it to people, I've been able to think less about the details of putting it together and more about the possibilities going forward from that, and how we can actually use that to make other people's bioinformatic analyses easier and more powerful. So that, I think, is what I'm looking forward to in the near term.